So first of all, our first presentation will be by Dr. Muhammad Ali Syed. Dr. Syed, the floor is yours. You may unmute and start sharing your screen, please.
First of all, I'd like to extend my warm regards to the great conference team. I hope I'm audible. My voice is, I hope, listenable to you people. I say hi to all the participants. I hope they will be able to provide good input to my next research. This was my recently published research work, if I believe that, and I've just listened from you that I have just 5 minutes. So I will just skip to the main part then in that scenario. So my topic was HPLC method validation for the estimation of lignocaine, HCl, ketoprofen, and hydrocortisone. So these are commonly used drugs, although used alone, but in combination, then normally we have to follow HPLC method. My respected juries present, they are quite experienced. So I'm just sharing my research to them. Normally, going to the introduction. So eczema is the situation in which inflammatory wound is a clinical disorder which is normally worsened by the presence of pathogens. So ultimate outcome is that we have to use multiple combination of drugs. So an important quality system in ongoing in the industry is that we have to check the quality as well as the quantity of the drugs that are passed through the industrial batches. So likewise, for such kind of clinical situations, we normally use a combination, although this combination is not used worldwide, like lignocaine, ketoprofen, hydrocortisone. The research says that either of the two combinations are present in the literature. So we just applied for the patent as well. For method development section, we use the current settings, HPLC instrumentation settings, because when multiple drugs are used, we have to go for HPLC instrumentation for analysis. So we initially created the method, then we validated it according to the guidelines, and eventually we try to check the safety of the method in terms of environmental hazard. So I'm just quickly skipping to the main part. So these were the materials available for my research. I just procured it from those industries. These were the instrumental conditions: Shimadzu machine, water company, Agilent column. Injection volume was 20 microliter. Important point relative to greenness was we used acetonitrile and phosphate buffer solution in equal ratios. So these were the system suitability and method validation protocols we used. Just going to the main part. Okay, so it was the outcome of the HPLC chromatogram, which shows that we successfully able to separate out all the active moieties present in our formulation. Lignocaine first, followed by hydrocortisone, ketoprofen at respective times of 1.5, 2.5, and 5.8. So in short, all the peaks were able to elute out before 7 minutes using the current ratio, 50:50. So previously in our pilot scale work, the organic phase was 70%. So we had to cut it down to 50%. We used in different combinations of the solvent, the aqueous solvent. The separability of the peaks were apart. So once a pH of 3 was used, and an equal volume of the organic as well as the aqueous phase was delivered, we were able to separate out the peaks before 7 minutes. Okay, so it shows the concentration we tested on the column with a mean area of plates. Theoretical contents were 40, 60, and 80%. System suitability parameters showed that the retention time was fine. It was, above 2. The tailing was less than 2. The resolution was better, above 4. Then we move to the accuracy. Accuracy was as well fine. Since we use the same concentration as used previously, the accuracy was almost above 99% with RSD value of less than 1%. So in pharma industry where testing is involved, normally we have an assumption according to the guidelines that an RSD value of less than 2% is considered safe and effective. I mean, problem free. Okay, so it was the amount that was delivered and that much amount was recovered. It was accuracy. So meanwhile, we worked on different days on different machines at different levels by the different analysts. And more or less, the recovery was better for the precision purposes because precision allows us to monitor minor changes in the system that are intended just to observe that what are the possibilities if work place machine is changed, or like if analyst is changed, what is the impact of this variation. So in our current scenario, again the accuracy in terms of precision actually was above 99% with the relative standard deviation was less than 2. Likewise, the linearity parameters showed a good regression relationship between the linearity concentrations prepared, and this is the linearity, 0.6 microgram per ml to 56. While for ketoprofen, we had to go for 0.2 to 100. For LOD, it was 0.563 for lignocaine. So on, it's displayed on your screens. If you can see, Lignocaine has LOD as well as LOQ. Then hydrocortisone has a value of 0.6 to 1.8 for LOQ. Because detection is the concentration which can be easily detected as the minimal value. While quantification, we consider it as a minimal value that can be considered to quantify the amount of the analyte. So in the validation parameter, last come was robustness. In robustness, we deliberately intended to have minor changes in instrumental conditions like temperature, pH, as well as the flow rate of the mobile phase in the system. Again, no doubt, the time was changed. There were slight changes, but overall output, or you can say in terms of accuracy, the contents recovered were more than 99 with the RST value less than 2% for each drug. So in terms of specificity for our current project, we actually use these two polymers. We prepared a solution of the standard solution of these polymers inside water, and that just had, I mean, after the SOP, it was filtered out using a millipore filter with a capacity of 0.3 micron. It was filtered out again. No peak was observed where the peak of these analytes were seen or has been reported as retention time. So just coming to our relevant portion. In current study, we applied Agree, although there are numerous softwares available online like HPLC 8, Capital. Although we used Agree software, it is freely available at university website of Spain. So I will show you my email if anybody is interested to use this software to analyze the greenness. You are most welcome to contact with me. So what specifically superior to this technique of evaluating greenness was that it used a number of evaluation parameters for the marking of the safety, and if any parameter had an alarming score that is out of the reach, and in simple words, if it is toxic to the environment that it actually used a penalty points. And when the penalty was given, so points were removed from the overall score. The overall score with a maximal value of 1 was considered as ideally green, while a value of 0 was supposed to be total failure. So a median response of 5.5 could be considered as an average score. So we used different questionnaire with specified options, while some of the questions were open while other were closed. For instance, in the question 1, it was sample damage. So we had to provide a response to a series of 12 questions, and where the score was poor, or you can say below to a failure level, it was pointed as 8. For instance, if it was a time consuming process, it would have given a low score. Similarly, if it was toxic to the environment, it was supposed to give low marks. Likewise, safety of operator was a valuable parameter. Use of energy, renewable sources, analysis per hour, waste amount, advertisation, transformation level, steps involved, location, sample size, sample damage. These were all the parameters that we have utilized and extracted from the information related to our developed method, developed and validated. So our total aggregated score was 0.75, which lied near to that black colored spot, which actually showed that our score lied inside the green contour. The green contour is considered as a bit more safer. I'm not claiming that it is the best method. So previously no method was reported, so we had to go for developing a novel method. So it is an open invitation for the researchers that if they could improve some of the conditions of our currently reported method, they are open. So what they need to do, they should similarly work on agri score and just compare their score, agri score, for the environmental safety for the newly developed method to us. So if their score comes out to below, so it means below 0.76, it means in terms of environmental safety, it was not appreciable. So our score, since for the estimation of 3 analytes in a single methodology, HPLC method, our score for environmental safety was 0.76. We also tested some of the marketed products locally. So we had to go for separate products. Since no semi-solid dosage form was present, and it was part of the semi-solid dosage form. So because we are the pioneer to develop a combination of these three drugs as a form of transdermal delivery for the arthritis patients, for the eczema patients, and for the patients who have some sort of unidentifiable skin etiologies. So the label claim proved to be best. Again, it was above the safer limit, and for that sake, it was identified as the commercial product as well at the same time. So the current conditions actually used a bit lower concentration of the organic phase previously reported in the pilot phase, which unfortunately, due to the shortage of contents, we were unable to share the data. So the current method used a 50% organic phase, the greenness of which found out to be 0.76. In terms of definitely the method development and validation, it was found reasonable, acceptable, accurate, precise, economical, robust, time saving in terms of 3 analyte in a single run. So it was a successful development of method, as well as its validation, its application for the validation of a marketed product, and eventually its safety evaluation using agri software. I just share some of the perspectives in terms of method development, but I mean, since I belong to a formulation scientist, Our group is now working on potential clinical outcome of the combination of these three drugs, so that we may be able, once the data is complete and finished and in the form of presentable work, we will definitely meet up again at some reasonable platform, hopefully at the grid. So currently it is in the stage of development of method and its validation using human plasma, as especially saliva of healthy volunteers. I'm definitely indebted to Selmore Pharma, my previous organizations that really supported me, my colleagues, my intern student who actually worked with me in terms of getting human pharmacokinetic data for the completion of that project, and all my well-wishers and students. This was the method that was published in the literature with this digital object identifier number. So this is my email address. If anybody has some issues or wish to contact with me in future related to object or for any new idea, he or she is most welcome. Thank you for your precious time.
Thank you so much, Dr. Syed, for your insightful presentation. Now, let's take the questions from the chairs and co-chairs.
Thank you very much, Dr. Muhammad. Thank you for this incisive contribution. And everyone in the room, you're welcome. Thank you for listening. I understand one of the greatest challenges analytical scientists face in the lab is the development of methods for simultaneous identification and quantification of organic chemicals for different purposes, either in the environmental space or in pharmaceutical formulations. So your methods, which have simultaneously identified and quantified 3 active ingredients, is indeed worthy of commendation. Please accept my appreciations for this. Just a few clarifications. The retention time for the first compound you had, which was Lignocaine, was at 1.554. I'm wondering, with subsequent injections, what were your findings on the robustness of this retention time? Did it shift in any way? Was it consistently within acceptable limits? That is, only the last decimal place was being changed, or there were any significant changes? Number two, maybe you can take two of them at the same time. You might have as well clarified whether this was a gradient protocol or an isocratic protocol. Which was this, please? Thank you.
Thank you, Dr. Bathur, for your question. Yes, definitely. I would like to respond to your second question. The conditions were isocratic, actually. But for the robustness, we had to fix to some single time point in terms of temperature, I mean to say. The method was robust, but we had to go for a single temperature point. And then from that standard point, we had to minor change the conditions. This was your, I believe, your second question, if I'm not wrong. Your first question was, yes, lignocaine is a drug. It is a local anesthetic drug. It is a bit tricky drug in the sense of being retenting out. Since it is highly water soluble, as well, it has the property of also soluble in most of the organic solvent. I mean, it has a dual behavior. So most of the drugs in pharmacy are water insoluble. For that sake, we have to increase the aqueous content. But this drug, which we have worked on, it was actually the drug which was I worked on in several projects. This drug has an issue that it elutes too early. Yes, I can understand this scientific behavior that a drug which elutes too early is not good, since the starting few Second, especially the first 2 minutes are settled for the noise or the peaks of the solvents that might be present inside. But we made sure that the peak that was present for lignocaine did not had any sort of noise. So there are many methods reported in the literature where the lignocaine eluted out even near to first minute, 1 minute, 1.2 minutes, 1.4 minutes. The reason behind this is it's too tricky. Its behavior. It can elute out if even we increase the organic content, it still can elute out. The reason behind its dual nature of solubility. So it is highly water soluble drug. So this is the problem that most of the researcher normally face. To overcome this problem, the only option left is to increase the organic phase too much. I mean, we are bound or we have to follow to increase the uh, ratio wise concentration of the organic phase. So previously we used 70% organic phase and 30% aqueous phosphate buffer. And even we had tried for 80-20 as well. But then we have to follow the integrity of the shape of the peak as well. I mean, I'm not talking about Lignocaine alone. We have to see the shape integrity of all three peaks. So for that sake, we found a ratio of 1:1 or 50:50 most relevant. Thank you. I hope you are satisfied with my response.
Oh, thank you so much, Dr. Said. Thank you. When you made subsequent injections, was the retention time consistent for this first eluting compound, lignocaine?
Oh, yes, definitely. We change different buffer systems while we were at pilot stage and when the peaks were not fully characterized for its shape. Once the conditions were fulfilled, yes, all the three peaks were consistent. Yes, in the robustness, there were minor change in the time, especially when we increased the speed of mobile phase, then the time was changed, which is actually logically sound for science that when you increase or decrease the speed of mobile phase, it has to come early or late. But for other accuracy levels, I mean precision, accuracy, linearity, the peaks were stable at where they actually stood. I hope it's clear to you, sir.
That's okay. That's good. Thank you so much.
So thank you so much, Dr. Syed, for answering Dr. Yarkwan's question. Now, I request our co-chair, Dr. Thakur, to put up her questions verbally.
Yeah, I have a question like in your research, sir, you have mentioned C8 column chosen instead of more common C8 for HPCL. So, did it give a better separation or peak symmetry? This is my question.
For C8s,
Yeah, C8 column. Why was a C8 column was chosen instead of the more common C18?
yes, C18 was as well. We just recently worked on two drugs, Ketoprofen and Lignocaine. It was on C18. But that was a different solvent system and a different scenario. But then when we added the third drug, which was hydrocortisone, it was producing peak overlap, after which we eventually had to go for some different type, I mean C8.
So, okay, fine. But do you think that is give a better separation or peak symmetry according to your research?
There could be a possibility that it could give better, but as shared that we used C18. We tried
Mhm.
some of the options that could possibly under, I mean, limited sources, but the only option left logical was C8.
Okay. Okay. Thank you so much.
Thanks, ma'am, for your response.
So thank you, Dr. Seth, for your presentation and for answering the questions of our chairs and co-chairs. You may stop sharing your screen also.